Yep, you didn't misread that. In this video, guys, we're going to go through the entire content for Biology Paper 1. That does not include the required practicals, but it includes everything else you need to know. Now, we're going to go at lightning pace, so make sure that you utilize the pause feature, pause things, rewind them when you need to, but we're going to go through absolutely everything in just over an hour, and so buckle up and let's get to work. Okay, so the objectives of this video, you need to be able to answer this paper 1 like an absolute champ. Also, you need to quickly highlight the areas in which you need more work, and we will try in this video to solidify that knowledge. If you need to go and do more work on it, because obviously this is going to be rather quick, then you need to be able to answer this paper like a champ. So that is exactly what we're going to do, and here we go. Okay, so eukaryotes and prokaryotes, right? Organisms can be separated into either eukaryotes or prokaryotes, and it's basically based on the types of cells. A eukaryotic cell has a nucleus. For example, we have eukaryotic cells. We are eukaryotes. Plants are also, so are fungi, etc., etc. Prokaryotic cells do not have a nucleus, right? All bacteria are prokaryotes. So let's have a look at those cells. A prokaryotic cell, in particular a bacteria, looks somewhat like this. And so, what do we have? Well, we have a cell wall, right? The cell wall on the outside. What does that do? Well, it stops the cell from bursting and it provides protection to the cell. We have the cell membrane, right? The cell membrane. Now, you should know what a cell membrane does. A cell membrane basically controls what is allowed to go in and out of the cell, right? Then, this stuff which you can't see, I'm not pointing at the white circle. The stuff you can't see is just the jelly-like cytoplasm, which is where all of your chemical reactions occur. Okay, this big white thing in the middle is the genetic material, right? The genetic material. Sometimes it's called the bacterial chromosome, right? It's basically the DNA of the bacteria because there is no nucleus, right? It's just floating around in cytoplasm. These things I've drawn the same color for a very good reason. These are plasmids, right? Now, plasmids are independent pieces of DNA which are separate from the main chromosome. And finally, these dots are ribosomes, right? And the ribosomes make proteins. All right. Moving on, you have animal cells. Now, this is a better diagram. The one I drew before, obviously, is a bit more amateur, but almost all of the stuff here is the same. So the cell membrane, okay, a cell membrane obviously controls what goes in and out of a cell. Notice that animals do not have a cell wall, right? The ribosome does exactly the same thing as before. That makes proteins. The nucleus, well, the nucleus controls basically what's happening in the cell because it contains the DNA, right? It contains the DNA. Before, we just had the genetic material floating in the cytoplasm, right? That moves me nicely to the cytoplasm. Cytoplasm is where the chemical reactions in general happen inside the cell, aside from maybe one of them, which is aerobic respiration, which happens in the mitochondria, right? Individual is called a mitochondrion, plural, the mitochondria, but that is where aerobic respiration happens, and we'll look at that later on in this video. So, plants. Okay, plants. Their cells are slightly more complex, but not too much. They still have the same stuff. So cytoplasm, okay, that's where all the chemical reactions happen. The ribosome, they make proteins. The cell wall, just like in the bacteria, the cell wall stops the cell from bursting. It provides structure to the cell. Cell membrane controls what's allowed to go in and out. The nucleus contains the DNA. Chloroplast, right? The chloroplast is where photosynthesis happens, right? So this is the green stuff. It contains chlorophyll, right? I'll just spell that out for you. We're going to look at it a bit later anyway, but it contains chlorophyll, okay? So a chloroplast contains chlorophyll. A vacuole contains cell sap, right? Cell sap. Now, cell sap is basically sugary water, so it contains sugars and other minerals and water, and it provides structure to the cell. It also, obviously, is a sugar storage for the cell. Mitochondrion, or the mitochondria, etc. We know exactly what that does. Okay, so that is what your cells look like, but that is not what all of your cells look like. We have what's called cell specialization. Now, cell specialization is basically where we have cells which look different, they do different things, that they have different components, right? Our cells are actually able to specialize, right? Because they need to carry out a specific function. Okay, tissues are normally groups of specialized cells which all carry out the same function, right? And we're going to have a look at a few examples of specialized cells. Boom, so first we have the sperm cell, right, which is this one at the top left. The sperm cell clearly doesn't look like what the most, or what the generic animal cell looks like. Now, the sperm cell has a nucleus up here in its head, and it actually contains half the DNA, right? A sperm and an egg cell contain half the amount of DNA of the rest of our cells, obviously because they need to join together, and then that makes a full organism. Now, they have a load of mitochondria in their body, and then they have this tail, right? Now, the tail is so that the sperm can swim, because the sperm needs to make its way to an egg in order to fertilize it. Now, moving actually requires energy. The mitochondria is there so that it can carry out aerobic respiration and provide that energy. So that is why this sperm cell is shaped exactly like it is. So, root hair cell is the next one. Root hair cell. Now, I'm sorry for my bad writing. I'm going to try and make that a little bit better for the rest of the video. But a root hair cell. Now, the job of a root hair cell is to absorb water and to absorb minerals, right? We're going to have a look at this in more detail again in a little bit. But it has this protrusion, right? This protrusion, or the root hair, as it's actually called. Now, this actually gives it a high surface area to volume ratio, right? And having a high surface area actually allows diffusion to happen faster. It allows osmosis to happen faster. It allows basically things to be absorbed by that cell more quickly, and then it can pass it on to something else. Then finally, we have a neuron, right? This top right is a neuron, which is basically a nerve cell. Now, this is a very interesting cell. What it actually does is it sends electrical impulses, right, from the cell body away. And so electrical impulses are allowed to go down here, right? These things are called myelin sheaths. Myelin. And I just said I was going to make my writing better, and this is like even worse. So I am going to try from now on. This is called a myelin sheath. Now, a myelin sheath provides insulation, and therefore it allows electricity to be conducted more quickly. It's like the outside insulation on an actual electrical wire. That's kind of a way you can think about it. Then here, right, so all of these, and all of these, right, you can think of these as basically nerve endings. Okay, these actually join with other neurons, okay, because the electrical impulse needs to be passed from one of these cells to another one so that we can coordinate all the information. It can get to our brain, it can go back out of our brain to tell us what to do, etc., etc. By the way, this long part here, yeah, so all the way, is called an axon, right? So the electrical impulse travels down the axon, and that is how it actually works. Okay. Now, some important information about specialized cells is that once a cell becomes specialized, it's not actually then able to differentiate into another type of cell. Differentiation, or cell differentiation, is the term we use to refer to a cell becoming specialized, right? Now, plants actually retain unspecialized cells in a tissue called the meristem. Okay, now these can then grow into any type of cell. Once a cell has specialized, it can't then turn into another type of cell, but an unspecialized cell can. And so plants are able to do that. That's why we can actually cut off a bit of a plant, plant it in the ground, and actually grow a whole new plant. You certainly can't do that with a human. Wouldn't make sense to cut off part of your leg, put it in the ground, and then grow another human. Obviously, that doesn't make any sense whatsoever. Okay, so microscopes. There are two types of microscopes, if you are broadly speaking. Those are electron microscopes and light microscopes. Okay, an electron microscope actually has a higher resolution and a higher magnification than a light microscope, right? Just so you know, magnification means basically how far you can zoom in. Resolution is actually the distance between 2 points, or the shortest distance between 2 points, of which you can tell the difference. So, for example, if you can zoom into an image but it's really, really blurry and you can't actually see the difference between one point and another, then that's a lower resolution. If you increase the resolution, then you can actually tell the difference. A good example of this in real life, by the way, is something like a TV or a phone screen or whatever, right? The higher resolution means that there's more pixels, which means you can obviously tell the difference between one dot and the next one, and it makes it less blurry. But anyway, moving on, we have our light microscope, and you need to know this basic structure. So the eyepiece lens, right, this does magnify, but this is where you actually look, right? You've then got here the objective lenses, right? And the objective lenses, you can switch those, and they also magnify. So you've actually got two parts which magnify. Yeah, and the overall magnification is actually the magnification of the eyepiece lens multiplied by the magnification of the objective lens. Alright, you've then got two focusing dials. The coarse focus, right, that moves things quickly so that you can quickly move things kind of into focus, and then the fine focus allows you to tweak it a little bit and try and get it exactly as you want. What they actually do is they move the stage up and down, right? And the stage is basically this. It's where what you're looking at actually rests. Let me rub those out. Now, what you actually have here is a specimen. Now, the specimen is what you're actually looking at, right? And the specimen is basically under or between what we call a glass slide, right? And so that piece of glass is actually a slide. The specimen is what you're actually looking at, right? The stage is where it's sitting. Underneath, you have a mirror, and that mirror reflects light up through the sample, or through the specimen, through your eyepieces, so you can actually see. Now, sometimes you don't have a mirror there. Sometimes you have an actual light source instead, which is better. Okay, and this is the equation that I mentioned before. Magnification of the eyepiece lens times by magnification of the objective lens gives you the total magnification. For example, a lot of time the eyepiece might be a times 10 eyepiece. Okay, and your objective lens, where you have all different types, you might have a times 100, or you might have a times 50, or you might have a times 200, etc. Your overall magnification will be those two multiplied. Yeah, so 10 times 100 equals 1000. That would be your total magnification if you were using those two lenses. Okay. Now, another way in which you can work out magnification, which is the more common thing you're going to be asked, is if you have the image size and you have the actual size. So let's say they gave you a diagram of a cell and they told you how big that cell is in real life. Now, what you would do is you'd have a look at the size of the cell. Let's say that they told you the cell was 0.01 millimeters, or that's the same as 10 micrometers, right? And then you measured the image and it was like 2.4 centimeters. Right, now I'm going to write it in millimeters because those units have to be the same. So 2.4 centimeters is 24 millimeters, right? Now, if I was using my actual, my equation, I would take my image size, which is 24 millimeters, divided by the actual size. So the actual size of the cell, which is 0.01 millimeters, right? Because they're both in millimeters, that means that the units cancel out and we get our magnification. And your magnification in this case would be 2,400, right? And so it's 2,400 times magnification in that example. All right, so moving on, we mentioned electron microscopes. Now, electron microscopes clearly look different. They're a bit more complicated and they're a lot more expensive. And they can give you a higher resolution, a higher magnification, and they only give you black or white images. Black and white, sorry, not black or white. Black and white images. Okay, now there's two types, and those are the scanning electron microscope and the transmission electron microscope. Right, a scanning electron microscope is one in which a beam of electrons is used to scan across an entire sample or an entire image, okay, but not at the same time, and then those images are put together to give you an overall image. Now that can give you a 3D image. On the other hand, a transmission electron microscope actually fires electrons through the sample in one direction, and you end up with a flat 2D image, just like you would normally get from a light microscope, except it's going to be way more detailed because you have such a higher resolution. Alright, so now let's talk about your DNA, or just DNA in general. So DNA is organized into chromosomes. Each body cell has chromosomes in pairs, right? Which means that your cells are diploid cells, with the exception of the gametes, which are the sperm and the egg, and those have half the amount of DNA, and so we call those haploid cells. Okay, so this is basically the hierarchy of your DNA, right? Your DNA kind of has this structure, right, on the right-hand side. And then if you add loads of those together in a big long chain or long polymer, then you get what we call a gene, and these genes are organized onto chromosomes, which is what you see on the left-hand side. Now, a gene is a piece of DNA which basically codes for a characteristic, and there are loads of genes on a chromosome, right? And this on the right-hand side is just an image showing you an actual real-life representation of your chromosomes. Okay, so genes are sections of DNA which code for characteristics. They actually code for proteins, right? And those proteins give you characteristics. Each version of a gene, though, is called an allele, right? So you could have the hair color gene, but you have the blonde hair allele or the brown hair allele, right? And so that's the difference between the two. Okay, so the cell cycle. Now, cells are constantly dying and in the process of being being remade, right? Cells just don't exist for your whole life. You're always replacing your cells, and different cells are replaced at different rates. Mitosis is the type of cell division with which we replace our cells, right? So if we need to replace our cells, they divide by mitosis. Now, that is one part of the cell cycle, and the cell cycle has 3 parts in broad. So the first part is called interphase. Now, most of your cells are in interphase, right? Interphase is the normal functioning life of a cell, but while it is in interphase, it is growing and it's increasing the number of mitochondria and ribosomes and other things inside the cell so that it's ready to divide. Right? Then during mitosis, the cell does divide and it produces 2 identical cells, right? But then they need to split apart from each other, and that splitting apart is called cytokinesis, and then you end up with 2 separate cells. Right, so let's have a look. So you start off with a normal cell, right? What you do is you double the amount of DNA, and then those chromosomes actually pair up. Okay, they pair up, and you can see in blue and red that you have those chromosomes now in pairs. Okay, so the nucleus disappears during mitosis after this has happened, and those chromosomes, which have now been copied and paired, they actually line up in the middle of the cell, right? They line up in the center of the cell, as you can see here in the third diagram. And then what we have are these fibers, right? Those black things, so these, right, I'm just going to rub that out, are actually called spindle fibers, right? They join to each individual pair of chromosomes, and then they pull apart and they separate the 2 copies of chromosomes to either end of the cell. And so that's what you get here, and that's what you get here, right? Then the cell begins to divide, because if you think at either end of the cell, you now have a full amount of DNA that you had in the original cell. And so you separate those to either side, and then the cell starts to divide. Then cytokinesis occurs, right? Which is this stage. When you go from here and you actually split apart, that's actually part 3 in the cell cycle, which is cytokinesis. So interphase happens before this. This is mitosis, but this last part here is actually cytokinesis. Okay, stem cells. So stem cells are undifferentiated. They can differentiate into specialized cells. We've spoke about those already. Embryonic stem cells in animals can differentiate into anything, right? They're what we call totipotent. Okay, adult stem cells, which we also have in our bone marrow, for example, they can only differentiate into certain types of cells, and so they're not called totipotent, they're called pluripotent, right? Adult stem cells, like in your bone marrow, can differentiate into blood cells, right? They can form red blood cells, they can form white blood cells, but they can't form things like nerve cells or muscle cells, right? So that means they're not as good, if you like. Plant cells, though, contain totipotent stem cells in a tissue called the meristem. We spoke about that already. That's how plants are different to animals. That's why we can clone plants simply by cutting parts of them off. Right, so stem cells could provide cures, though, for various different conditions, because they provide the opportunity for us to actually grow new cells, right? New cells and therefore new tissues. For example, if you were a major burns victim, you could grow a load of your skin tissue to cover it up and then skin graft it, and then it's your own skin growing from your own stem cells, etc., etc. Okay, we could actually replace whole organs, etc., and so that's very interesting. But we could also use them to genetically engineer a human being and change their DNA, and obviously there are ethical issues behind that, right? People get really, really upset about this kind of thing, and so you have to be careful when you're answering questions. You need to add up both sides, right? Well, there's clearly potential benefits from it, but there's a chance it can be abused. Also, where do you get the embryonic stem cells from? If you're taking stem cells from an embryo, then you're probably killing the embryo, and some people argue that those embryos are already living things, living people, and therefore you're actually killing people, and so some people actually see it as murder, and so there's massive ethical issues behind it. Okay, moving on. Transport. Transport is just the movement of stuff. You have 3 types of transport you need to know, which is diffusion, osmosis, and active transport. Diffusion is the net movement of particles in a fluid, right? That just means not a solid, so it could be a liquid or a gas, from a high concentration to a low concentration down its concentration gradient. And this is an example, right? This is in the lungs, right? So in the lungs, this actually happens. Now, these are alveoli, right? So this is your alveoli. Alveoli. Okay, and this here is our blood. Right, it's our blood vessels. Now, when we breathe in, we get oxygen into our lungs, into the alveoli, and then oxygen diffuses down its concentration gradient, because high concentration here, low concentration here, into the blood, and that's exactly where we want it. Right, on the other hand, we've got a load of CO2 in the blood and not in the alveoli, and so that diffuses the other way, and then when we breathe out, then we get rid of that carbon dioxide, and then the process continues. Okay, osmosis. Osmosis is kind of like diffusion, but it only includes water, and so it's from a high concentration of water to a low concentration of water, and it must be across a partially permeable membrane, right? That's so important. Has to be across a partially permeable membrane. Now, what a partially permeable membrane actually means is just a membrane which lets some things through and doesn't let other things through, okay? So, for example, it lets water through but doesn't let, like, large proteins through it, which a lot of membranes don't. Okay, so what would actually happen in osmosis? Well, it depends on the concentration of water in your two solutions. Now, just to be frank, if you have pure water, then that's the highest concentration of water that you can get. And as soon as you start dissolving other things in water, its concentration goes down. So the more things you have dissolved in water, the lower the water concentration, okay? And water will follow its concentration gradient. So what you have here is three scenarios. So we have a cell, and this round the cell is the membrane, which is a partially permeable membrane, cool. And then outside it, we have another solution, right? So we have two solutions, the inside of the cell and the outside of the cell. In scenario A, we say that both solutions are isotonic. Isotonic means that they have the same water concentration. And if they do have the same water concentration, then the net movement is going to be zero because the concentrations are already the same. B, the outside solution is known as hypotonic. Right, hypotonic means it has a higher water concentration. So water moves from a hypotonic solution into the other solution. Okay, and so it moves down its concentration gradient. On the other hand, right, by the way, the blue dots obviously are water. And in C, it's the other way around. You have a hypertonic solution, which means the concentration of water outside the cell in C is actually lower than the water concentration inside the cell, right? And therefore, water moves from the cell and the cell shrinks. And this is actually the basis for one of your required practicals, the one with the potatoes, which we'll look at in a separate video. Okay. Active transport, so movement of particles from a lower concentration to a higher concentration, which requires energy, right? That part is so important. This does not happen naturally. Osmosis and diffusion happen spontaneously, naturally. You don't have to do anything. Active transport does not. Active transport, one example of active transport is using the root hair cell, which is a type of cell we've already seen. They carry out active transport to bring in even more solutes, even though the cell maybe has more solutes than the outside does already, right? So the point is that the plant wants as many salts as possible. It might be magnesium, might be potassium, might be nitrates, etc. And if they already have a load, they still want more, and so they bring in the extra using energy, right? However, using energy is important, right? It actually does use energy in order to do this. It doesn't just happen naturally. And so they need to carry out respiration in order to provide that energy. Okay. Deep breath. Next topic coming straight up. And so, organization levels in living things. You need to know this hierarchy of organization. So, the smallest basic unit of an organism is the cell. Cells or similar cells are grouped together in tissues to carry out a function. Those tissues are then grouped together into organs to carry out an overall function, right? And then organs are grouped together in organ systems to carry out a broad living function. And then organ systems are grouped together, and that forms your overall organism, which is actually the thing, right? For example, we are organisms. Okay, so one example of an organ system is our digestive system. The purpose of it is that we need to absorb nutrients from our food, right? This allows us to break down large insoluble bits of food into small soluble molecules, which we can actually dissolve, absorb, and then actually use. Okay, so small molecules can actually diffuse into the blood and be transported where they're needed, but the large insoluble molecules actually cannot. And so here is our digestive system. Let's talk through the individual parts and their functions now. So first of all, the salivary glands, what do? Well, they make saliva. That seems blatantly obvious. But what's the point in saliva, right? Saliva actually acts as a lubricant, right? As a lubricant. Okay, so it allows us to swallow things, etc., etc. And also, it contains amylase, right? Amylase, which is an enzyme which breaks down carbohydrates, right? So an enzyme which breaks down carbohydrates. That's why if you put something that's a carbohydrate into your mouth, it'll eventually break down and dissolve in your mouth. But something like a piece of steak, which is made of a load of protein, actually won't. All right, you then have your esophagus. Basically, that just connects your mouth to your stomach. And so the esophagus, that's pretty much its only job. The saliva helps things travel down the esophagus and reach the stomach, and the stomach is where a lot of the good stuff happens. So the stomach basically churns your food, right? So there are muscles in the wall of the stomach which churn your food and break it down into smaller and smaller pieces, which allows us to digest it. Now the stomach also contains pepsin, right? Pepsin, which is a protease. So it breaks down protein. So actually your food is beginning to be digested in the stomach as well, right? It also contains stomach acid, right? Stomach acid. And that's because pepsin is an interesting enzyme. It works best at pH 2 or 2.5, right? Most enzymes work best at pH 7. We're going to have a look at this in a bit more detail in just a little bit. But the stomach acid has another job. Also, if you swallow pathogens, right, if you swallow things which give you disease or whatever, they actually get killed in the stomach because of the stomach acid, and so that is great. All right, what about the liver? Well, the liver actually produces bile, right? It produces bile. And bile is really important because the stomach acid would actually damage any other organ in your body. And so bile neutralizes the stomach acid to make it okay again, right? It returns the pH to an okay amount. What bile also does is it emulsifies fats, which just means it allows us to dissolve fats and therefore absorb them as we move on into the small intestine. The gallbladder, by the way, is just where bile is stored. So you make it in the liver and you store it in the gallbladder. Okay, the pancreas. Okay, the pancreas actually produces protease. Right, protease is a broad category of enzymes which break down protein. Carbohydrates, which actually break down carbohydrates, and lipases, which break down lipids, which are fats and oils. Now, these enzymes are actually produced in the pancreas, but then they are sent off into the small intestine because they actually work in the small intestine, not in the pancreas. The pancreas' job is actually just to produce them, right? So the small intestine, that's where a lot of the good stuff happens. The small intestine is actually not very small. It's actually the longest part of the whole digestive system, right? It's really long, but the job of the small intestine is to absorb all of this good stuff that we've been breaking down into the blood. Right, so the small intestine is where you absorb all your nutrients and stuff into the blood. And notice on this diagram, we've got these things called villi, which is actually the lining of the small intestine, right? The small intestine is actually lined with all these stuff, right? The walls are not smooth. They look like this. That is to massively increase the surface area, which massively increases the rate of diffusion. Now, that is extremely important because it means that we can absorb things from our food before they end up being released. Perfect. And then once that has happened, right, so you've absorbed things like amino acids, glucose and stuff like that, and fatty acids and whatever from your fats, then everything that hasn't been digested basically goes into the large intestine. And the large intestine is basically where your insoluble stuff like fiber, okay, is, and you absorb water from it. And then once that's all done, then the last of your food gets to your anus, and then you know what happens after that. You get rid of it, and that's called defecation. Alright, so we mentioned digestive enzymes. Let's have a look at them in a tiny bit more detail. You've got carbohydrates, which breaks down carbohydrates into sugars. Amylase is an example. Proteases break down proteins into amino acids. Pepsin is an example in the stomach. Lipases break down lipids into fatty acids and glycerol. You don't need to know an example of those. So how do enzymes actually work? Now, an enzyme is this blue thing, right, which is just a big protein. And it's got this part here, which is called the active site. Right, the active site is basically where something fits into it. The job of an enzyme is just to speed up a chemical reaction, right? That's all it does. It's called a biological catalyst. But what really happens is what's known as the lock and key hypothesis, right? You have something which just fits into that enzyme, and therefore it allows the enzyme to work on it, and then the reaction can happen, right? So for example, in a digestion, if you have a carbohydrate, then a carbohydrate is that orange thing which just fits into the blue thing, which is the enzyme. And then it's able to be broken down, and you see on the right hand side you now have your products after they have been broken down. Now, problem occurs if you either have a temperature that's too high or a pH which is too different to the pH that the enzyme likes. Enzymes have specific temperatures which they like to work out. Enzymes have specific pHs which they like to work out. And you can see here that if the temperature or the pH changes too much, then the enzyme changes shape, right? This is called denatured, right? This enzyme has been denatured, right? It doesn't mean the enzyme is dead, but it means it won't work anymore. Enzymes can't be dead because they're not alive in the first place, right? But it won't work anymore because it's irreversibly changed shape. And notice that this molecule now cannot fit in it, and that is that. Okay, so let's have a look in more detail. Temperature. Most enzymes have an optimum temperature of around 37 degrees. That's why our body temperature is 37 degrees. They can't go too much higher, right? So if this is actual activity of the enzyme, or its rate of enzyme reaction, okay, you can see it's got an exponential curve as it increases, because rate of reaction just increases as temperature goes up. But then if you go too high, then this happens and you get your enzyme denaturing, and then it plummets and you get no reaction at all, right? At like 50 degrees or whatever. That's why if our body temperature reaches 50 degrees, we are guaranteed dead. Okay, so pH. pH is different. pH, it's not just that the enzyme... Sorry, if I go back, just quickly, notice that the reaction is not 0 at 0 degrees. That's because even if you freeze the enzyme, it doesn't mean it's denatured, it just means the rate of reaction is really slow, okay? If you freeze it, it's not denatured. If you go too hot, it has denatured. So back to pH, if your pH goes too high or too low, both will denature your enzyme, and therefore it's broken, and the rate of reaction just goes to 0, okay? And so that is the graph that it looks like. If we had something like pepsin, by the way, which has an optimum pH of around about 2.5, the graph would look exactly the same. I say exactly the same, like I can't draw, but in a different place, right? So that's basically it. You have enzymes which work best at different temperatures, just most of them are pH 7. Okay, so the heart. Moving on, the job of the heart is to pump blood to our lungs to be oxygenated, and then pump that oxygenated blood to the rest of the body. We have a double circulatory system, whereas fish and insects have a single circulatory system. So let's take a look at the structure of the heart now. So this is the heart. It's actually the reverse orientation. So what you see as the left is actually the right. It's as if you're looking at someone who's facing you. So this here is the right side, and this here is the left side, right? So what you have is 4 chambers in the heart. The top 2 are called the atria, singular atrium, and the bottom 2 are called the ventricles. Yeah. And so first, blood comes in to the right atrium, right? The job of the atria is to take blood in, right? Then they contract and pump blood through to the ventricles. So blood comes into the right atrium from the body, right? Not from the lungs, from the rest of the body. So it's deoxygenated, but comes in here. Then it gets pumped into the ventricle, right? The right ventricle. And then the right ventricle pumps blood to the lungs via a blood vessel called the pulmonary artery. Now, any vessel that leaves the heart is called an artery. Any vessel that comes back to the heart is called a vein, right? So the pulmonary just means the lungs. So it's pumped via the pulmonary artery, right? That actually goes to the lungs and gets its oxygen and does whatever it needs to do. Then it comes back via the pulmonary vein. So the pulmonary vein brings blood into the left atrium. That pumps into the left ventricle, okay? And then that pumps via the aorta, which is this one, to the rest of the body. Okay, so it comes into the right atrium. So deoxygenated blood, let's just rub this out. Deoxygenated comes into the right atrium, pumped to the right ventricle, then pumped to the lungs. Then it comes back into the left atrium, gets pumped to the left ventricle, then gets pumped to the rest of the body, and then it happens again. The reason it's called a double circulatory system is because it enters the heart twice in one cycle. So if you want to have a quick go, look at this diagram which has no labels, pause the video and have a go at labeling it, but in the interest of time, I'm going to move straight on with blood vessels. So there are 3 types of blood vessels: arteries, veins, and capillaries. The arteries carry blood under high pressure away from the heart. The capillaries, which follow from the arteries, carry blood to the tissues and to all your cells, and they actually are what is involved with the exchange of things like oxygen and whatever. And so they are really small, and they go around all of your tissues. Then finally, the blood, it ends up in the veins, and they're kind of the motorway that brings blood back to the heart so that it can repeat the process. And so here we go. On the left-hand side, you have an artery. You see it's got these thick walls, right? The really thick walls. It's got muscle in them as well. And the lumen is actually quite thin, right? There are no valves in the arteries either. Because it's under high pressure, it doesn't need them. And because it's under high pressure, that's why it needs those thick walls so that they don't actually burst, right? They lead into the capillaries. The capillaries are really small. They're actually 1 cell thick, their walls. And so that allows them to quickly allow things to diffuse in and out, which is exactly what they need to do because they're exchanging stuff with other cells. And then finally, the blood leaks into the veins, and then the veins take it back to the heart. By the way, the veins, the reason they've got thinner walls and a larger lumen is because it's under a lower pressure, right? It's not being pumped into the veins, it's being pumped into the arteries. And so it's a lower pressure. They need valves to stop the blood from going back on itself, right? And those valves allow the blood to go in one direction back to the heart and everything to be okay. Okay, so what is your blood actually made of? It's more than just the red blood cells. It actually also contains platelets, white blood cells, and plasma. The red blood cells are actually what makes your blood red, right? They look like this. So on your right hand side you can see red blood cells. Okay, they've got this flat biconcave disc shape which gives them a high surface area. That's great because it allows them to transfer oxygen, right? That's actually their only job. They contain a load of hemoglobin, yeah, and that hemoglobin actually carries oxygen. Yeah, they actually don't contain a nucleus. They don't contain mitochondria because those things would just take up space. Instead, they're just packed to the brim with hemoglobin, which allows them to carry a load of oxygen. Okay, platelets are actually small fragments of red blood cells, right? They're small fragments. You can see them on the bottom right, and they act as like a mesh to stop you from bleeding. They all clog together and form this mesh, stops you from bleeding, and they get joined together by a protein called fibrin, which is great. The white blood cells, they're involved in your immune system. We're going to have a look at those in a little bit. But phagocytes and lymphocytes are two examples. Blood plasma is actually the liquid part of your blood. And this is the part everyone forgets. Your blood is a liquid, right? Those red blood cells are not the only part of your blood. Actually, the majority of it is plasma, which is yellow in color, right? It's only red because we have the red blood cells. If you separate them, you do get the plasma, which is yellow. It's got a load of water in it. This is where things like sugar dissolves, right? So your blood glucose concentration is actually because it's dissolved in the plasma. All right. And so finally on the heart, we have heart disease, and coronary heart disease is one example of that, so I put it in brackets. Your coronary arteries are the arteries which actually take blood to the heart muscle, right? Not going into the heart, into the chambers, but actually supply the heart with oxygen. And if they get clogged up, then your heart could lack oxygen and lack energy, and therefore it could stop working. So that's a severe problem. That's what causes heart attacks, right? And one of the main reasons is they get clogged up with fat like this. Right, it looks all gooey and disgusting, but this is actually what happens if you get a load of fat deposits in your arteries. And this is actually called atherosclerosis, right? So and I'm going to have to write fairly neatly. Sclerosis. Okay, that is when you have fat deposits in your arteries, and that can actually lead to really high blood pressure, can lead to vessels bursting, can lead to heart attack, etc., etc. We can actually try and heal those by using these things, which are metal cages called stents, right? Those stents open up the artery, which is great. So what else? Well, we can also replace faulty valves. If you have a faulty valve, it can cause blood to mix where it's not meant to mix, and that can end up with you not having a high enough blood pressure. It can end up with you not having blood pumped around the body properly. And so we can replace those with either other valves from other animals or from other humans who have died, or we can actually make our own out of carbon fiber, and we can use those. Finally, if your heart, which is able to produce its own electrical impulses, it actually causes itself to beat, right, on its own. If the part of the heart that does that, so your own natural pacemaker breaks, you can use a man-made pacemaker, which is what you can see here. That actually supplies your heart with electrical impulses which allow it to beat as it should. All right, moving on, we have non-communicable diseases. Now, coronary heart disease is a non-communicable disease, but we have other ones as well. Those are ones which cannot be transferred from one person to another, especially by a organism. Okay, so obesity is another example of that, right? Poor diet and exercise is the general cause of obesity. Coronary heart disease, as we just saw, is an example. Now, there are several risk factors for non-communicable even disease. And those are things like alcohol, those are things like smoking, those are things like eating really fatty, sugary foods and ignoring eating your 5 a day. The opposite to that is eating a really healthy, balanced diet and getting regular exercise. Those drastically decrease your risk of non-communicable disease. And so that is something really important to remember. However, one example of a non-communicable disease is cancer. Right, not a very happy topic, but you do need to know about it. So cancer is a result of changes in cells which causes them to uncontrollably divide. That causes a tumor. Now, a tumor can either be malignant, and then a malignant tumor is the bad one or the really bad one, where it grows out of control and spreads from one part of the body to the other. In order to treat that kind of cancer, you need to find it and stop it really early, right? It is doable, but it's something that is difficult. You need to find it and treat it early on. If you're late in that stage, then it's very very difficult to treat. A benign tumor, right? So a benign tumor is actually normally contained within a membrane, which means it can't spread out of its particular location. Those ones are generally less serious, but they can still be serious. For example, if you have a benign tumor in the in the brain, then that obviously causes problems. And so here is just a diagram showing you these cells here are forming a tumor, right? They're just multiplying and multiplying, and eventually you're going to get more of them and more of them and more of them, blah, 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 blah. The cells around it are normal cells, but you can see that they're getting outgrown, outcompeted by those cancer cells, which is not a good thing. So what causes cancer? We just said that mutations in DNA or changes in your cells, which hint is a mutation in your DNA, causes it. So it's actually caused by mutation in DNA leading to that uncontrolled cell division. More than 20% of cancer has been linked with smoking and or alcohol consumption, right? That is quite a startling percentage. But when I say linked, it doesn't mean that they have definitely caused it, but it's like, well, they've probably caused it, yeah? Because mutation can just happen randomly. You can have someone who lives the healthiest life in the world, never smokes, never drinks, and still gets it, which is annoying, right? But the chances are just lower. Other things, ionizing radiation can cause mutation, right? If you're near something that's radioactive, then that can actively cause mutation, which can lead to cancer. Genetic factors can cause mutation, right? If you've got a certain gene, the BRCA gene is a very common example for breast cancer, and also being infected with something like the HPV virus or hepatitis B virus, those can increase your chance of cancer as well. Okay, so how do we treat cancer? Well, we use chemotherapy, right? Chemotherapy means using chemicals, and you use them to actually kill the cancerous cells. And we use radiotherapy, and that means using radioactivity in order to kill those cells. The problem with these are that they are not cancer specific. They will actually kill any cells, and so if you target them to the cancer cells, you may also be killing, and you normally are also killing, healthy cells as well, and so that can do damage. And depending on where the cancer is, you might want to be more careful. So if it's in the brain, then you've got to be extremely careful, right? Healthy lifestyle is a great way of treating it. It's actually a great way of avoiding it, but also stopping it from growing faster and faster, right? So make sure you live that healthy lifestyle. Finally, antibodies, right? Antibodies are a very interesting, more recent treatment for cancer, and that's something we're going to have a look at a little bit later on, but not yet. Okay, so moving on to plant tissues. So plant tissues, right, we just spoke about animal tissues. Plant tissues, we're going to be a bit quicker with. Here you have a cross-section of a leaf, right? This is like looking at a leaf sideways. And you've got all these different layers of different cells, which are actually grouped into tissues. So you've got the waxy cuticle, which is right this bit at the top, which provides basically protection to the leaf. It's not water. It doesn't let water in, right? It's waterproof, and so that's why you see water dropping off the top of the waxy cuticle. It doesn't really let water out either, which is a great thing. The upper epidermis, which is basically a protective layer as well, not really much happening there. Palisade mesophyll, right? Mesophyll, basically you can think of that as where the good stuff happens, where the photosynthesis happens. The palisade is the main one, right? You see these are big cells which are all bunched together, and so there's loads of chloroplasts, and you've got loads of good stuff happening there. Spongy mesophyll, right? Those are towards the bottom. They still carry out a lot of photosynthesis as well, but they've got holes in between them. That's why they're called spongy mesophyll. There's a reason for those holes being there. It's so that oxygen and carbon dioxide can be exchanged and let out without going into another cell. Finally, you've got the stomata down the bottom, right? Those are actually holes, right? So each one is called a stoma. Overall, they're called stomata if there's more than one. Those are holes which the plant can open and close. Around them, right, I didn't write this yet. Around them, those are called guard cells. Right, I'll actually write that. Guard cells. Guard cells. And what they do is they actually open and close the stomata. Right, that's their job. Okay, so xylem and phloem. And so if we have a look on the left-hand side here first, you have the phloem. Now, phloem is responsible for moving food, basically glucose and sucrose, around the plant, right? It's basically made up of cells which are interconnected, right? And you've got these sieve tubes between them, right? And this in the middle is pretty much, let me use a different color actually. This in the middle is pretty much hollow, right? But these cells are actually alive, yeah? So these move food around the plant. They have next to them companion cells which look more like normal cells. Those basically have the nucleus, and those do the normal cellular things. What's in the middle actually lets things go through. It's like a pipe. Yeah. Now on this side you have the xylem, right? The xylem. Now the xylem looks very similar, but the job of the xylem is actually to carry water and minerals. So what you absorb from the soil, from the roots up to the leaves, and it's only in one direction, right? That's important. Phloem can actually carry things in any direction, right? It could go this way, could go up, could go down, etc., etc. Right? The xylem only carries it. The xylem only carries the water and the dissolved minerals to the leaves from the roots, right? So it's in one direction. Okay, and xylem cells are fully dead, right? You can see there's no nucleus. You can see that it doesn't look like you've got all this stuff going on like you do in the phloem. The xylem cells are dead. The phloem cells are alive, and that's another big difference. Okay, so transpiration. Transpiration is basically how water and those dissolved minerals move throughout the plant. So it all starts off in the roots. Remember we spoke about root hair cells taking in water, right? And obviously taking in anything that's dissolved in the water as well, okay? Then what happens is the water goes from the roots up the xylem, right? Like so, and then into the leaves. Okay, that's the basis of what happens. But why does it do that? Okay, you can think of this as sucking through a straw, right? But it's actually called the transpiration stream. If we go back to our leaf diagram quickly, yeah, what's happening quite a lot is water and oxygen and carbon dioxide, right, are lost from here, right? Mainly water and oxygen. Carbon dioxide is actually taken in quite a lot, but it can be released as well because of respiration. But we're losing a load of water, more importantly, from the leaf, right? If that stomata is open. Well, when that happens, it creates a pressure difference. It is like sucking through a straw, right? Because it's connected, right? You've got the transpiration stream. It's basically connected to the xylem vessel, which is going up the stem. And so when the water leaves, it creates a pressure difference, which causes water to come up to replace it. Okay, so the water actually then comes up. If this wants to work, then that would be great. It then comes up the stem in order to replace it, right? And then down here, the roots actually then absorb more water, and then it happens again. More water is then lost from the leaves, and it happens again. And so that is the transpiration stream. Now, what affects how fast that happens is basically diffusion, right? Diffusion occurs for water to leave the leaves, right? To leave the leaf, or to exit the leaves, if you like, into the air. And this is a really common question. Most people will call it osmosis. It's not osmosis because it's not happening through a partially permeable membrane. It's actually diffusion, right? And there are 4 things you need to know that affect how fast that's happening. One is wind. If it's really windy, then it moves water away from the leaf, which increases the concentration gradient, and so it happens faster. If it's really humid, right, then there's load of water outside the leaves. If there's a load of water outside the leaves, then the concentration gradient is lower, and so it happens slower. If the temperature is really hot, right, then it just happens quicker. Diffusion happens quicker. Also, it means that the plant has probably opened its stomata, which increases the rate of diffusion of the water out, which increases the rate of transpiration. And finally, light has kind of the same effect. It causes the stomata to open. The plants open their stomata when it's light, and that increases the rate of transpiration as well. So all 4 of those things increase the rate of transpiration. And that is about as far as we're going to go with plants. So now let's have a look at communicable disease. Alright, so what is a communicable even disease? Now this is a disease which can be passed on from one organism to another, and they're caused by microorganisms, right? And we call those microorganisms pathogens. Now they can be things like viruses, fungi, bacteria, or protists, and we're going to go into those in a second. A pathogen infects a host, reproduces inside the host, causes damage, and then infects a new host, right? That's how they get around, that's how they spread. Pathogens are adapted in many different ways to carry out their functions. There's 2 examples here on the right. At the top, you have a drawing, really, of what a virus looks like. And then at the bottom, you have a real-life example of fungi, which have infected a plant in this case, and have sprouted a load of hyphae. So, moving on. How do we actually spread disease? Well, there's a few ways. Now, if we look in the middle, many are actually airborne, right? So, airborne. Now this means that they can actually be transferred via the air. If you cough or you sneeze, or even if you just breathe and talk, then you can breathe out these pathogens and they go and then infect something else. Many of these are actually viruses, but they don't have to be viruses, okay? Okay, so how else? Well, this one here, dirty water, right? If you drink dirty water, or even if you just take a bath in dirty water, etc., and then put your hand to your mouth, you could ingest those pathogens. Cholera is a good example of that. Okay, direct physical contact. So this handshake is an example of direct contact. If you haven't washed your hands, or if you just come into contact with a pathogen, you shake someone's hand, then they've got it, and either they put their hand to their mouth or whatever it is. Okay, another one is contaminated food. This is an obvious example of that, right? Some bacteria or whatever it's going to be, or fungi, actually sprouts in the food and they grow in the food. When you eat them, then you are now sick. Finally, they can be transferred by another animal or a vector, right? This is called a vector. The reason I've put a mosquito is because malaria is actually caused by a microorganism, which is a protist called Plasmodium, but it is actually the mosquito that allows transfer from one organism to another. Okay, so let's have a look at some examples of viruses or viral diseases. Okay, first we've got measles. Now, I've actually spelled that wrong. There's meant to be an A there, so that was silly me. Measles is a virus. Okay, measles is something which is very, very common, and it is an airborne virus, right? You generally get it when you're a kid because it is very easy to transfer. However, once we have been infected once, normally you don't become infected again, and so that's why you generally are going to get those once. Okay. What about HIV? Now, luckily, HIV is not as common. HIV is an STD, so it's a sexually transmitted disease, and it's extremely bad. There's no current cure at all. Treatment has gotten a lot better, but what HIV actually does is it blocks your immune system from working by actually attacking your immune cells, right? This is really bad, and it means you probably die of something else, like a different kind of sickness, and so that's not good. TMV stands for tobacco mosaic virus. Now, this is actually a virus which infects plants, right? Which makes it interesting. Because it affects plants, what it actually does is it affects the leaves mainly, and you get all these black kind of spots. That reduces the plant's capability of photosynthesis and therefore stops them from getting so much energy, and therefore they'll eventually die. Okay, so bacteria. Bacteria, two examples here. You've got salmonella. Salmonella, this is actually a kind of illustration of salmonella, but this is what it looks like. And salmonella you can get via your food. It causes sickness. It causes severe vomiting and stuff like that. And generally it's spread via like the handling of food without washing your hands or something like that. And so that's pretty bad. Okay, gonorrhea. Gonorrhea is another STI. It's a or STD or STI, disease or infection. It's a very, very common STD, but it's not very nice. It is a bacterium. What it actually does is it causes a yellowy discharge from the penis or from the vagina, and that is obviously not very nice stuff. It also makes urinating painful, and so that's also not very nice. Okay, so fungi. You have what's called rose black spots, right, which infects plants. It causes these lesions which are blackish or rose blackish, if you like, and that inhibits the growth of plants because photosynthesis is affected because the parts which have been infected can't carry out photosynthesis. The leaves eventually drop off early, and then eventually the plant will die if it's not treated. You can treat it. You can actually treat it with fungicides, and then you're okay. Athlete's foot is a common fungal infection in humans. It is caused by a fungus, and it is a problem because it causes irritation, it causes your feet to smell, and it can actually lead to worse conditions if you don't treat it. It's spread by contact, like if you walk in a bathroom with your feet off and you've got it, then that's a way that it can be spread. So in swimming pools is a very common way of passing it on. Okay, protists, right? Malaria is caused by a protist, and let me just circle it here. This here is the Plasmodium. Plasmodium. Okay, and that is actually the pathogen, right? It is a protist, and what that means, or what that is a small single-celled microorganism, right? But it is different from viruses, it is different from bacteria, etc., etc. But Plasmodium doesn't just travel around in the air on its own. In fact, it uses a vector, and that vector is this guy over here, or I say guy, it's actually a girl. It is a female mosquito. Now, female mosquitoes actually take in these Plasmodium, and then when they go around and suck blood, they can give you that pathogen, which is not good. Malaria is a very serious condition. Okay, but how can we actually stop them? Well, that is a great question, which I'm going to answer right now. So the first line of defense is the skin, right? The skin is a great organ. It covers your entire body. It insulates you and protects you from pathogens, right? The skin is constantly dying and replacing itself. So your outer layers of skin fall off, and then you produce new skin on the inside. And so that is something that's constantly happening. It also produces antimicrobial compounds which fight pathogens and stop you from growing lesions of things on your skin. So that's all well and good. Okay, so tears and saliva contain enzymes which kill bacteria, right? Those enzymes are called lysozymes, so that's something useful. Mucus is also produced, right? So when you have phlegm and stuff like that, that is actually mucus. What it does is it's really sticky and it traps pathogens inside it, and then we can swallow them. Once we swallow them, they're in the stomach. Well, the stomach is full of stomach acid, and stomach acid kills all those pathogens, and that's all good. Right, second line of defense are the phagocytes. We talked about these earlier a tiny bit. They're basically one type of white blood cells. What they do is they engulf things, which actually means they eat things. They kind of find the pathogens, and they take them in, and then they just basically digest them inside, right? They digest them just like we would digest our food. They take them in and then they digest them and kill them. Okay, so but they're not specific. This is why we call them the second line of defense. They're not specific. They'll go around and they'll pretty much engulf anything which is not from us. So they won't go around and engulf our normal healthy cells. But if we take in anything else, so any other cell, and they find it, they'll just engulf it. And so everything is fair game in that sense. Third line of defense are the lymphocytes, and this is actually specific. So the lymphocytes are white blood cells, and they produce antibodies. Antibodies are proteins which have specific shapes on them, right? They bind to stuff. They bind to what is known as the antigen on a pathogen. Okay, so if we look here, you might not be able to see it. Let me just actually zoom that in. If we look over here, this is our pathogen. So this circle thing, this blob, might be a bacteria. Well, these are the antigens, right? These little shapes on the end are the antigens. Now, they have specific shapes, and all cells have these things on the outside, right? All things have these on the outside. And basically, your antibody or antibodies can be complementary to those. So they will go and basically bind them. And so they are specific in that sense. Well, why do we care about that? Well, if they bind them, then it basically can clump pathogens together, so it stops them from going around and doing what they need to do. Also, when they are bound to an antibody, it actually causes the phagocytes, so these guys, to go and eat whatever they're bound to, and so that is good. Now, it so happens that we actually maintain an immunological memory of anything that we have produced antibodies for. So we are randomly producing these antibodies all the time, and most of them aren't going and binding anything. But if by chance one antibody does find a pathogen and bind to its antigen, then we remember that, right? Which means in the future, if we come across that antigen again, we can produce the antibody straight away. Otherwise, it's just by chance, right? We're producing different antibodies all the time, and eventually one of them is going to be complementary. But during that time, you might be infected by that pathogen. But once you remember it, you're going to respond to it way quicker. And that is actually the reason for vaccines. So vaccines actually contain dead or inactive pathogens, right? Vaccines are obviously injections. Our body reacts as if they were real infections, and it produces antibodies or lymphocytes for them, right? Antibodies from lymphocytes, right, for those. And so that means that eventually you have a memory of that antigen or / that pathogen, but you're not getting infected by it because they're actually dead or they're inactive. But it means that if you come across the real thing another time in real life, you already have the antibodies in order to attack it. So that is great. Okay, so how else do we treat conditions? Well, one of the big ones is antibiotics, right? Antibiotics. So these are antibiotics. An antibiotic will kill bacteria. It won't kill viruses. It won't kill fungi, right? You have antifungals for funguses, or for fungi, sorry. You have antivirals, which kill viruses, but those they kind of vary in how good they are. But the problem is that bacteria can actually become resistant to antibiotics. That is a very, very bad thing, right? That's why... Okay, and I've represented that with this picture down here. That's why we don't use antibiotics unless we need to. Okay, if you have an infection, let's say you have a cold, right? Colds are generally caused by viruses. If you take antibiotics because you think it's going to help, well, one, it's not going to help. Two, it means that you could have other bacteria becoming immune or becoming resistant, to that antibiotic. Okay, that means in the future, when you do have a problem and you take an antibiotic, you actually got bacteria which are resistant to it, and that's not a good thing. Okay, so limit the amount of antibiotics you use. Also, take them in combination. So take more than one different antibiotic at a time, drastically reduces the chance that you have any bacteria become resistant. Okay, something else important is the painkiller. Painkillers are great because they kill pain, right, as the name suggests. But they do not actually treat the condition whatsoever. Instead, all they do is they alleviate that symptom. So if you've got a headache and you take a painkiller, great. You're not actually curing the headache, right? Whatever's causing the headache, you're not actually doing that, but you are stopping yourself from being in so much pain, and so that is a good thing, but you can't use them to actually cure yourself of a condition. Okay, so drug development. Importantly, drugs take a very, very long time to be developed, right? And so most potential drugs never make it to market because the testing is so strict and it takes so long. The first thing that anyone does is computer modeling, right? They basically model what the drug is going to do in real life, but in a simulation. Okay, they model it so that nothing's being harmed, right? Next, once they've got something which they think is going to work, then they take it into a lab and they test it on cells, and then maybe they test it on animals as well. So that might be mice, it might be flies, it might be whatever, right? Generally, if it's a drug that's going to work on humans, you want to use a mammal, so mice is a good one. Finally, if all those are passed, then you move on to human trials. Now, the first phase is a small dose on healthy individuals, right, on healthy volunteer individuals, and that's to test for side effects. Secondly, if that is okay and you don't get horrible side effects, then you use a small dose of it on actual patients to test efficacy, which just means how effective it is, and dosage, right? Because if you don't know exactly what dose to use, then you need to do these tests. Finally, a larger pool of patients is tested to finalize that dosage and to make sure it's effective. Yeah, and during those trials, they are generally blind trials, which means that some of the people are actually given a drug which isn't the drug. It's just like a tablet with nothing in it. And then you can test for the differences between those people and the people who are actually given the real drug. And if there's a big difference, then great, you think that the drug's working, right? Generally, you're going to use a double-blind trial. That's really important because it means that the people who are looking at the results, so the doctors, they don't know who was given the drug either. All they're doing is looking for differences in symptoms before and after taking whatever they've taken. Okay, so monoclonal antibodies, right? If you are not doing triple science, you're not doing biology, you may just ignore this part, maybe fast forward a little bit. But a monoclonal antibody, or a group of monoclonal antibodies, there we go, are antibodies which are identical, right? They're produced by fusing a mouse spleen cell with a myeloma cell, right? That is a cancer cell. That forms what we call a hybridoma. Now, those antibodies are great for detecting things. We use them in the pregnancy tests, right? They test for HCG, which is a hormone, and so that is all well and good. They can also be used to target drugs in a very specific way, right? They can target parts of our body in very specific ways because they are very specific themselves. They can go and bind to something. We can tell them to go and bind to a certain type of cell, for example. However, research on that kind of thing is still ongoing. You do need to know how we produce them. Well, a mouse is first injected with an antigen, right? And the antigen, obviously, is what you want your antibodies to detect or to attack or whatever it is. And then you fuse the spleen cells of that mouse, which has been injected, with myeloma cancer cells, and you form what we call hybridoma cells, right, via a cell fusion. Okay, then those hybridoma cells, okay, they are going to grow your antibodies, and so you harvest them or you culture them in a medium that allows them to survive, but it doesn't allow other cells to survive, because all you want is those cells that have actually fused. What you would also have is the spleen cells and the myeloma cells which haven't fused, and you don't want those. So you make sure that you only allow those to survive. Then you take out the antibodies, and that is your product done. Okie doke. And so finally, this is pretty much the last section, right? We're going to have a look at photosynthesis and respiration. So photosynthesis is a chemical reaction, right? Green plants and algae do it. They turn carbon dioxide and water into glucose and oxygen, right? Using energy from sunlight. This is an endothermic reaction. Endothermic just means a reaction in which energy is taken in from the surroundings. So here are the equations. Water plus carbon dioxide in the presence of sunlight, that's my great drawing there, makes glucose and oxygen. And I've given you the simple equation. Learn that if you want to. Otherwise, don't bother. And so, here we go. This on the left is algae, and that on the right is a plant. I don't need to tell you that. By the way, what would surprise you is that most of the oxygen that's produced via photosynthesis in the world is by algae, not by plants, because overall, you have a massive volume of algae where there is a water supply. Okay, so rate of photosynthesis. The rate of photosynthesis, right, so how quick photosynthesis actually happens is limited by several factors. Those being temperature, carbon dioxide, sunlight, and chlorophyll, right? So the amount of chlorophyll they've got. Because remember, chlorophyll is what absorbs the sunlight, and so they need that. Okay. You need to know the shapes of the graphs or the relationships between those factors and the rate of photosynthesis. Now, this is very important, right? If it's limited by these factors, then these are called limiting factors. Limiting factors, right? So, first of all, rate of photosynthesis versus carbon dioxide concentration. As you increase the carbon dioxide concentration, the rate of photosynthesis will increase until, finally, you've given it enough carbon dioxide that it flattens off, right? What does that actually mean? Well, it means at that point, something else is limiting the rate of photosynthesis. Okay, that's what that really means. It means you've got enough carbon dioxide that maybe one of the other things, so sunlight, is limiting photosynthesis at that point, right? So at this point, you've got so much CO2 that now, if you increase sunlight, you're gonna increase the rate of photosynthesis instead. And so there's the graph for that. Surprise, surprise, it looks exactly the same, right? The light intensity, or the amount of sunlight, as you increase it, the rate of photosynthesis increases until that is no longer the limiting factor, and now something else is limiting it, right? Well, what's that something else? It might be the amount of chlorophyll, right? And so there you go. There's the same relationship again. As you increase the amount of chlorophyll, the rate of photosynthesis is going to increase as well. Obviously, you can't just add chlorophyll and increase it, right? You're actually talking about the amount of chlorophyll in the plant. And so that's actually quite a difficult experiment to do, if you like. But those three things have kind of the same relationship to the rate of photosynthesis. What doesn't is temperature. Okay, now you probably recognize this graph. It looks identical to the shape of the enzyme graph that we looked at earlier, right? That's because it is identical. Because photosynthesis is a bunch of chemical reactions which are controlled by enzymes. Okay, so that means that when it's really cold, the rate of reaction is really, really slow, but it doesn't mean that it doesn't happen at all, right? It happens, but it's just not a lot. Now, as you increase the temperature, the rate of reaction increases quite exponentially, right? It is a curve. And then, when you get to like 50 degrees, it goes all the way down because enzymes get denatured at that point, right? You have an optimum temperature for enzymes, which is going to give you the fastest rate of photosynthesis. After that, it starts to break down, and that is that. Now, photosynthesis produces glucose. What do we use glucose for? Or what do plants use glucose for as well? They use it for respiration, right? Remember, aerobic respiration is how organisms obtain energy. Also anaerobic respiration, but respiration in general. They obtain energy by releasing the energy stored in glucose. Okay, you can also use glucose, put a load of glucose together and turn it into something else and you'll get fats. Right? Also, you can add glucose to nitrates to form amino acids. Those amino acids can then be used to make proteins. Okay, but you need to have nitrates, hence why plants need nitrates. Also, glucose is just put together to make starch, right? Starch is obviously the storage molecule of glucose in plants. It's not in animals. In animals, it's glycogen, but in plants, it's starch, right? They add a load of glucose together to make starch, and then when they need energy, then they break the starch back down again. On the other hand, cellulose, if you put a load of glucose together, but in a different way, so in a different shape, you actually make the insoluble carbohydrate called cellulose. That makes the cell walls, and so that is why you use it. So, respiration. We're going to move on now as a quick another topic into respiration. So respiration is not the same thing as breathing. This is one really common misconception, right? Respiration and breathing are not the same thing. Respiration is a chemical reaction which releases the energy stored in the glucose, right? So photosynthesis makes glucose. Respiration releases the energy in the glucose, right? They're kind of the opposite of each other. It allows us to carry out processes which require energy, which is great. And so here are the word equations again. Handwriting is a lot better this time. So glucose plus oxygen gives out energy, making carbon dioxide and water. And there is the simple equation again. I don't have to read that out to you. Aerobic respiration requires oxygen, right? And occurs in the mitochondria in cells. Our cells carry this out all the time, right? It's not like photosynthesis where it can only happen in sunlight. We're always burning energy, right? Our cells are always doing something. We're always moving. Even when we're asleep, we're moving, right? Because you're breathing. And all of that good stuff requires energy, and so we're always carrying out respiration. The reaction is exothermic as it releases energy, right? Which is the opposite of photosynthesis. The more energy we need, the more glucose and oxygen we need to allow respiration to occur. And this is why we eat and why we breathe, right? There's the difference between breathing and respiration. You breathe in order to get oxygen, in order to use it for respiration. They're not the same thing. Okay, anaerobic respiration. Well, that is, or sometimes it happens, sorry, when you're out of breath, right? If you're sprinting, or you can't get enough oxygen, etc., we start respiring anaerobically. That's because you don't have enough oxygen. This produces lactic acid, which causes us to fatigue and cramp up. It doesn't last long. That's why you can't sprint for very long, right? Glucose actually forms lactic acid. That's the whole equation of the reaction, and you give out energy, right? Just like in any other respiration. However, anaerobic respiration only gives out a small amount of energy, 5% of the energy of aerobic respiration, so it's not as good. Anaerobic respiration, right? When you have finished, you need to get rid of the lactic acid. Well, the way you get rid of the lactic acid is you react it with oxygen. For that reason, when you are full of lactic acid and you've just been sprinting and stuff, you will be breathing really, really heavily, and that's so you can get extra oxygen, right? And you keep doing that until you've recovered. The amount of oxygen you need to clear all that lactic acid is known as your oxygen debt, right? Your oxygen debt is the amount of oxygen you need to clear your lactic acid. That also, by the way, releases the rest of the energy, which is great. Anaerobic respiration is different in plants and yeast, though. They carry it out in a different way. They don't produce lactic acid. Instead, they produce ethanol and carbon dioxide, right? We call that process fermentation. So when you probably heard of it, whenever you hear about things fermenting or fermentation, it's actually the anaerobic respiration in plants or in yeast. Useful for us because we can use the CO2 produced to cause bread to rise, and we can also use the ethanol produced to brew beer or make wine, right? And so that is a good thing. Okey-doke. By the way, we can't use that ethanol to produce things like vodka and spirits because we don't get a high enough percentage out of fermentation in order to do that. Okay, so finally, metabolism. Metabolism is the sum total of all the chemical reactions that happen either in a cell or in an entire organism, right? So these reactions include digestion, respiration, protein synthesis, etc., right? So all the chemical reactions you can think of, add those up, and that is metabolism. Some reactions do not require much energy. Typically, those are breakdown reactions, right? For example, respiration actually releases energy rather than uses it. Others typically do require energy, ones which make things. So synthesis reactions typically require energy. Okay, so breakdown reactions including breaking down proteins, right? Protein breakdown. Amino acid breakdown, so breaking amino acids down into like glucose and into some other stuff. And lipid respiration, so burning lipids in order to produce energy, right? Those do not require energy. However, synthesis reactions, where you make stuff, including glucose being converted into starch, into cellulose, and in animals being converted into glycogen, those require energy, right? Those are synthesis reactions rather than breakdown reactions. Lipid synthesis involves making fats from fatty acids and glycerol, requires energy. Protein synthesis, right? Glucose and nitrates are used to make amino acids, and then amino acids are put together at the ribosome to form proteins, and that is also a synthesis reaction that requires quite a lot of energy. And so, deep breath. That is actually it, right? That is all the content that you need, aside from the required practicals, which is going to be covered in another video. That is all the content you need for paper 1 of biology. I hope you've managed to keep up with that. I've had great fun just talking as fast as I can and ruining my voice as a result. But I hope that has really helped you guys. Please feel free to put any comments in the box below or send me a direct email. As usual, please like and subscribe to the page because that really does help me out. Obviously, follow me on Instagram and all that good stuff as well because that would be great. You'll be obviously notified of any new videos that come available, so it's useful for you as it is for me. But I look forward to seeing you in the next video.